Today's presentation is Mechanistic Model-Informed Discovery and Development of Bispecific Antibodies for Immuno-Oncology. It takes two to tango. Our speakers are Dr. Chi-Chung Lee and Dr. Pete Van de Graaff. Next slide, please. Chi is currently a principal scientist at Genentech, where she has more than 13 years of clinical drug development experience. She earned her doctorate in chemical engineering from the University of Florida. Pete is currently a senior vice president at Certara. He's also a professor of systems pharmacology at the Leiden University in the Netherlands, and I don't know how he manages to do all these things and do them so well, but he's also editor-in-chief of the scientific journal Clinical Pharmacology and Therapeutics. Pete and Chi, welcome to today's event. I'll now turn it over to Pete to begin the presentation.
Thank you very much, Suzanne, and welcome everyone. It's my pleasure to kick this webinar off with a brief introduction to bispecific antibodies and the role of quantitative pharmacology and mechanistic modeling in bispecific drug development. So let's start with the basics and a simple definition of what a bispecific is, namely an antibody that can bind to two different antigens at the same time. Although they are relatively new therapeutic modalities, bispecific antibodies have been known for many decades and were first reported in the early 1960s by Nisonov and co-workers. And here you see some of their seminal papers. However, as shown in this history chart, it took some 50 years from the first report of bispecifics to the approval of the first bispecific antibody for human therapeutic use. The initial steps and milestones were mainly fueled by advances and breakthroughs in biotechnology and antibody engineering, whereas more recently, biology, translational medicine, and of course, clinical pharmacology have played an increasingly important role in the development of bispecific therapeutics. In the last decades, this resulted in the launch of bispecific antibodies as novel therapeutic modalities in diseases of high medical needs, and here you see a few firsts. Catumaximab was the first bispecific antibody approved in 2009. 6 years later, the CD3-CD19 bispecific blinatumomab followed in the US. And in 2018, amikizumab followed as the first non-oncology bispecific for the treatment of hemophilia A. So let's pause for a little quiz now, for which I will hand it back to Suzanne.
Thanks, Pete. So we're going to test your knowledge of the folks on the call. So we just want to know if you can take a guess at how many bispecific antibodies are currently in development. Is it 30, 100, 300, or 1,000? We used pharmacology standards in picking these answers. So we'll just give folks a few more seconds to put their guesses in, and then we'll let you know if you guys got it right or not. All right, well, it looks like most people voted, so I'm gonna close the poll. And then let's share the results. So most of you said that it was an equal split between 100 and 300 bispecific antibodies in development. I'm gonna pass it back to Pete to tell you the real answer.
All right, thank you. So as you saw, we gave you options in steps of half a log order, which as clinical pharmacologist, of course, was meant to mimic a typical dose response. And well, here's the answer. So most of you actually got it right. So the current global bispecific pipeline has now over 300 entries. And it's predicted that in 5 years' time, the market for bispecifics will exceed $10 billion US. To talk about bispecifics as a single class is probably not doing justice to the huge diversity of modalities, since there are now more than 100 different formats, and the number keeps growing all the time. Indeed, trispecifics are already in development, and I think we will soon have to start to adopt the term multispecifics to cover this class of therapeutics. With the increasing diversity of formats, it is also no longer possible to assign a single overarching mechanism of action to bispecific antibodies. Broadly speaking, the mechanism of action can be classed as either in trans or bridging cells, such as T cells, NK cells, other immune cells, or in cis, which refers to bridging receptors expressed at the cell surface. The most exciting properties of bispecifics are called obligate, since these are properties that depend on the physical linkage of two specificities and cannot be obtained by combining separate antibodies with the same specificities. So simply speaking, these are the properties that differentiate the therapeutic potential of bispecifics from a combination of two individual antibodies, bridging the same targets. An example that will be discussed later is avidity, or functional affinity, which is the accumulated strength of multiple affinities summed up from multiple binding interactions. These unique or obligate properties of bispecifics clearly offer some great potential advantages, but also provide significant drug development challenges. For example, how do you select a safe and efficacious clinical starting dose? Since, as we'll show you later, the MABEL concept, which is well established for conventional monoclonal antibodies, may not always apply. Next, how do you define the efficacious dose to guide translation from preclinical models to human, and design a safe dose escalation study in early clinical development? Third, bispecifics have their own specific safety issues. For example, Cytokine Release Syndrome, which we will discuss in detail in this webinar. Last but not least, how do we predict and manage between patient variability, which, due to the multifactorial mechanism of action, may be more pronounced for bispecifics than what is typically observed with conventional monoclonals. It is important to remember that the variability is a combination of variability in drug properties, such as affinity, avidity, and pharmacokinetics, multiplied by the variability of the system parameters in each individual patient, such as target expression, cell expression, amount of soluble targets that can act as a shedding compartment, and target turnover rates. As a rule of thumb, you need to multiply this complexity and variability by at least 2 or more, if you take into account the obligate properties discussed earlier. So that brings us to the role of quantitative mechanistic modeling in the development of bispecifics, which will be discussed in detail by Chi after me. Briefly, as we outlined in more detail in this recent review in clinical pharmacology and therapeutics, mechanistic models are a key tool to manage the key development issues for bispecifics, like dose selection, I mentioned earlier, in particular in the context of the complex pharmacology and between patient variability I discussed in the previous slide. I think most people would agree that it is almost impossible to answer these key questions and therefore to develop a novel bispecific therapeutic without a quantitative pharmacology model. At Certara, we support many bispecific drug development programs for our pharma partners by developing bespoke models like the ones shown here, and with the biologic module embedded in the SimCip simulator software, An example of application of such models is to predict whether or not bell-shaped concentration-response relationships may be observed in early clinical dose escalation studies. I don't need to explain to this audience that it is, of course, of critical importance to know at which side of the peak you are when searching for an optimal and safe dose. The second example of the use of quantitative mechanistic modeling in bispecific drug development is Cytokine Release Syndrome, or CRS, which, as I mentioned, is arguably the most important generic safety issue for this class. If you have not read this already, I would highly encourage you to read the excellent review by David Feigenbaum and colleagues, which came out recently in the New England Journal. There are a few examples of quantitative systems pharmacology, QSP, models in this field. For example, this poster presented at a recent ACOP meeting by Regeneron. But it is still an emerging field. Arguably, this paper by Genentech is the current state of the art in the public domain, and Chi will discuss this in more detail. At Certara, we have recently developed PBPK models for 14 cytokines and linked this to our extended immuno-oncology platform. We think this can be applied in a unique manner to provide an integrated view of drug-induced cytokine release in the context of efficacy and safety. And we will be more than happy to discuss this with you after this webinar. So now I would like to hand it over to Chi, who will present how the principles of mechanistic modeling I have just summarized have been applied in drug development programs at Genentech.
Thanks, Pete. Good morning, good afternoon, everybody. Thanks for taking time to join the webinar. I hope everyone is doing well. It's a pleasure to present today. What I'd like to talk about today is some of the quantitative pharmacology work using mechanistic insights for mosunetuzumab, which is a bispecific antibody in clinical development at Genentech Roche. And model-based insights, in this case, helped us develop a dosing strategy which successfully reduced cytokine release syndrome. And the learnings, I believe, are broadly applicable to T-cell engaging bispecifics. So today I'll actually focus perhaps a little bit more on the application side and the pharmacology learnings supporting the mechanistic modeling. So mosunetuzumab, or Mosun in short, it's a bispecific antibody. One arm of the bispecific antibody targets CD3 that's expressed on T cells. The other arm targets CD20 that's expressed on B cells, and it's in development for treatment of B cell malignancies. And it is a full-length IgG1 antibody, in contrast to fragment-based bispecific entities, which requires constant infusion of drug. Mosun can be given with a more convenient schedule of once to every 3 weeks, given the longer half-life. And also in contrast to CAR T therapies, which require ex vivo T cell manipulation and therefore adds to the turnaround time for product availability, Mosun instead is off the shelf and readily available for administration. The mechanism of action for Mosun is illustrated here. And similar to what Pete had already mentioned, Mosun works by the dual binding construct, one arm to T cell and one arm to the target cell. This brings the B and T cells together, which creates an artificial immune synapse that activates the T cells, which then leads to release of cytotoxic granules, such as granzyme perforin, which causes tumor death. So as such, Mosun, in terms of mechanism of action, is a conditional agonist for T cell activation, which does not require full receptor occupancy to achieve maximal effect. And that actually is an important point to mention, and as you'll see, that plays in that role in our understanding of exposure response. So as Pete mentioned, cytokine release syndrome is a major safety concern across T-cell engaging bispecifics and CAR T cells, and it is an on-target effect. And specifically, one gives a bispecific antibody, it triggers the formation of immune synapse that activates the T cells, leading to a cascade of immune activation, resulting in cytokine release syndrome. So pharmacologically, one would expect the higher the dose, the higher the target engagement, leading to higher safety risks. So a key challenge for developing these agents have been to figure out a way to mitigate CRS while getting to an effective dose level to maximize the therapeutic benefit. And one way to do so is through a so-called step-up dosing regimen, which I'll show how it works in the next couple of slides. So to understand CRS, it's important to understand IL-6 and its kinetics. We know that patients with CRS tend to have higher IL-6 levels on average, so it's a useful biomarker to draw inferences to CRS risks. And before we had any clinical data for Mosun a couple of years ago, we wanted to leverage what we learned from preclinical data as well as what's published in the literature in designing our phase 1 clinical trial. And our goal at the time was to design a dose-finding strategy while mitigating CRS. So we collaborated with the Translational Systems Pharmacology group. They built a systems model shown in this figure that describes T-cell activation and associated IL-6 release in a couple of key physiological compartments. And this work is recently published by Husseini et al., and you can find more details in this publication shown at the bottom. But briefly, since the mechanism of action requires both the T and the B cells to work, the model describes B cell generation, maturation, and from the bone marrow, and also exit and traffic into other tissues. It also describes T cell generation from the thymus into the blood, and T cells can also traffic into different tissues. Excuse me. Sorry, technical difficulty there. And in all compartments, the T cell activation is described as a function of concentration of the bispecific antibodies, the T cells, the B cells, and this is the upstream driver for IL-6 release. So this model was then used to predict IL-6 profile, comparing different dosing scenarios. And for illustration purposes, I'm showing a fixed dosing regimen of 20 milligram every 3 weeks on top. And at the bottom, it's the same 20 milligram target dose, but preceded with a small step dose of 1.6 milligram a week before the target dose, so-called step-up dosing. And as you can see from the prediction on the right, the model predicts that step-up dosing results in approximately half the peak IL-6 level compared to the Q3 weekly regimen. So in other words, the model predicts that you can get to the same target dose level, in this case 20 milligram for illustration, but with only half the peak IL-6 level, which presumably leads to less safety risks expected. So the model importantly also points out an important kinetic feature of IL-6, where it is primarily a first dose effect, and repeat dosing seem to result in minimal modulation later on. So in this case, as you can see, even in the step-up dosing, with increasing the dose from 1.6 to 20, the IL-6 peak remained after the first dose only, excuse me. So here, this brings me to my first polling question to all of you. Knowing what we have just learned at a high level from the QSP modeling in terms of the dosing regimen and the kinetic feature, how would you apply this knowledge in a phase 1 study design? So A, informing the starting dose, B, inform the dose escalation step, C, predict the efficacious dose, or D, inform the dosing regimen. So I know you might have the tendency to want to select them all as a confirmed scientist and a pharmacometrician, but if you get to only prioritize your main impact in one area, what would it be? So Suzanne and Pam, please feel free to show the results when the voting is done. All right, thanks. So it seems like most people inform the dosing regimen. And I think, and some of you also pick starting dose and dose escalation step and efficacious dose. So I think really these all are good and viable options in my opinion. And obviously the model can be used to predict any of this. And I think it's useful sometimes to think about the area of most added value, and it really depends on your own unique molecule, excuse me, strategic context, depends on the key questions from your own team. And in our case, the primary impact was on B and D, so the dose escalation step and informing the dose regimen to be tested, phase 1. So I'll show you in more details how it was done. I'm trying to advance my slide. Sorry, I'm not able to advance my slide. Suzanne, can you help?
Yeah. Oh, there you go.
Thanks so much. Thank you. All right. So here's how we put the model prediction into test. This is our first in human study. It's a phase 1 clinical trial in relapsed refractory non-Hodgkin's lymphoma patients. In this study, we decided to test 2 dosing schedules. Group A is the IV fixed dosing group. Mosun is given every 3 weeks at the same dose level. Group B is the IVs, cycle 1 step-up dosing, where Mosun is given with 2 small ascending doses weekly, followed by the target dose on day 15, and then cycle 2 day 1 again, and then every 3 weeks. So since we learned from the modeling that the first dose is critical in determining the overall safety risks, we started with group A, escalation, and data from this group allowed us to build a robust dose-response relationship for safety and help determine step dose levels for group B. In group B, we ended up incorporating 2 step doses, primarily for extra safety precaution, as the context at the time was that it was the first CD20 bispecific antibody given in this patient population. And in addition, the second step gave us an additional lever to mitigate cytokine release syndrome, and we get to pivot based on what we see in patients. So as for the dose escalation, group B was escalated with dose window specific escalation rules. And this is a novel design in its own way because it allows us to cap the first 1 and 2 dose levels low based on observed clinical safety and the occurrence at the time window at which it occurs, compared to an internal safety target profile, while continuing to escalate the target dose level to achieve maximal response. So in a nutshell, what the model enabled us to do was to articulate and align on the dosing framework and across functions and with leadership, while enabling a design that informs clinical dose determination based on the empirical clinical data that we see in the trial in an efficient and informed manner. So pharmacologically, one way to understand this kinetic behavior of IL-6 and CRS that I just showed you is as shown here. The target engagement, as I described before, it results in T cell activation, both in the peripheral blood and in the tissue. The peripheral activation contributes directly to the cytokine release in the blood compared to the tissue activation, which is to a smaller extent due to tissue to blood partition. In other words, the peripheral activation is postulated to play a primary role in driving the IL-6 peak following the first dose. However, upon repeat dosing, since the targets are depleted by the drug, there's no longer many targets left that are immediately accessible from the blood. So at least in terms of what's immediately accessible from the blood, so leading to a much lower IL-6 the second time around. While this may not be the only mechanism at play, as we will discuss later, and there's no doubt that the understanding of the target kinetics and the accessibility of targets over time by bispecific in tissue can add great insights into understanding the CRS time course and its risk. So here is the clinical data, which really is exactly what we had expected. On the left, we're showing the PK, and this represents by the serum mean concentration of Mosun over time. It's colored by dose groups. As expected, the PK accumulates and increases with increasing dose level following the cycle 1 step-up dosing regimen. And in contrast, on the right, the IL-6 profile peaked primarily following the first step dose and remained relatively unmodulated upon repeat dosing, despite the escalating dose levels. So we again see the unique IL-6 kinetics that changes with time upon repeat dosing. And what about clinical CRS? And first, what's not shown here is the onset and the time course of CRS, but indeed it's primarily a first cycle effect. So that's consistent with the IL-6 kinetics. But what we're showing you here is the frequency of CRS and the exposure response. So here we're comparing again the group A fixed dosing schedule on the left versus the group B cycle 1 step-up dosing schedule on the right. On the y-axis, we're plotting the probability of a patient experiencing grade 2 or higher, excuse me, CRS. And on the y-axis, on the x-axis is actually the maximal receptor occupancy level, which for practical purposes, think of it as a model-based exposure metric representing peak target engagement level. And the dots here are observed clinical data. 0 represent no event, 1 represent yes. And the blue trend line is the logistic regression, while the numbers shown here are the observed CRS rate in each tertile of exposure. So overall, we can see that in group A, there is a trend of increasing CRS rate with increasing exposure. And that's even in the very small dose range we tested in group A, up to only 2.8 milligram dose. However, when we switch to group B, using the step-up dosing, despite going to more than 20-fold higher dose levels, the CRS rate did not appear to increase significantly. And it's very consistent with the modeling expectation. If we keep the step dose low, we can continue to escalate the target dose level without incurring much additional CRS risks. And note that even in group B, there is a slight upward trend seen at the high exposure level as we continue to push the target dose higher. But clearly, the exposure response curve has shifted to the right with a reduced potency for CRS using the step-up dosing schedule. And importantly, clinically, at all step-up dosing group cohorts, CRS is mild and transient, and there was no MTD reached across 3 orders of magnitude of dose ranging. So I want to take a brief moment to just mention that layering on top of the pharmacology story is another mechanism which we now know perhaps is also at play in addition to the target kinetics. So this is a work recently done by our research colleagues at Genentech using a HER2 T-cell dependent bispecific. And there may be an additional mechanism that has to do with T-cell desensitization in terms of cytokine release, such as TNF-alpha, and that desensitization triggers reduced potency also for production of IL-6. So as you can see this in this figure, upon repeat dosing, the potency for T-cells to release cytokines really largely reduced and leading to much less systemic cytokine to cause clinical CRS. And importantly, Importantly, this can be done without impacting the killing capability. So in other words, we start to decouple the drivers for safety from that for efficacy upon repeat dosing. So to put it all together, really the bispecifics works by engaging the T cells and the tumor cells to create an immune synapse to activate the T cells, which drives tumor killing. And it also leads to a cascade of immune response to produce IL-6, leading to CRS. And the integrated picture of the therapeutic window can then be represented by exposure-response relationship, where we see an increase in efficacy with the exposure that eventually plateaus at high exposure levels, as shown by the blue curve. And I'll show you the efficacy data for Mosun in a little bit, and we can comment on the monotonic increase then. But there's also an exposure safety relationship, but with repeat dosing, the relationship shifts to the right, reflecting reduction in potency for CRS. So the reason why step-up dosing works is by keeping the first dose low on the exposure response curve, where the CRS risk is most potent, and then escalating the target dose later when the ER curve is shifted to reduce potency. So this is how step-up dosing can expand the therapeutic index of T-cell engaging bispecifics. So that leads us to the second poll question. So now that we've essentially mitigated the on-target toxicity concern, here's another question for all of you. So how would you select the clinical dose based on phase 1 data set? So again, you can only pick one. Would you dose to a maximum tolerated dose? Dose based on perhaps a C-trough being greater than some in vitro benchmark, for example, EC90 from in vitro assays. Do PK/PD biomarkers, because oftentimes that can provide really useful insight in phase 1. Or would you conduct exposure response analysis for clinical efficacy endpoints? So pause a few moments for you to vote. Oh, okay. So it seems like everybody likes the idea of doing exposure response analysis directly on the efficacy endpoints and also PK/PD biomarkers. And then there are a couple also voted for a dose to maximum tolerated dose. And again, I think all these are good and viable options. Oftentimes, one might choose dosing to maximum tolerated dose just to not leave any efficacy on the table, especially for oncology development. In our case, we did not do that because in this case, we had a vision that we wanted really tolerable safety in order for the bispecific to be used in a community setting. In our case, any of the 3 other B, C, D, we did them all, but I'd say primarily what drove the dose selection in the end is probably D, so exposure response for clinical efficacy endpoints. And as you can imagine, doing that in a phase 1 trial where data is still premature and with limited sample size and lots of patient level heterogeneity, it really requires some level of practicality in modeling approaches, and probably relying on the strength of totality of data is usually a good idea. And also given that the premise is always with a heavy focus to speed up the development. So I can show you what some of the pharmacology story for how we determine the clinical dose. I'm trying to advance my slide. Sorry, Suzanne, can you help me? I'm not able to advance my slide.
Pam, can you advance it for Chief, please? There you go.
There you go. Thank you. I got it. Okay, so now switching gear to efficacy. So as you can imagine, our goal is to identify the switch to the plateau of the exposure response curve. So trying to identify a maximum efficacious dose. So when we first look at the clinical dose response, we were very puzzled. Here, what I'm plotting in orange is the overall response rate based on clinical objective response criteria. And in purple, orange is the complete response rate, and purple is the overall response rate. This is summarized across dose levels horizontally, and apparently we didn't see anything clear in terms of a dose-response relationship, despite having a robust more than 500-fold dose ranging in the clinic at the time. And the usual caveat of a small sample size, excuse me, there we go, a small sample size didn't really fully explain it either, because even when we pulled data across those groups using PK as a polling metric, we still saw no trend whatsoever. So we started thinking, really, what are we missing? Is there a confounder that we should be adjusting for in order to reveal the intrinsic exposure-response relationship we expect? So we took a step back and thought more carefully about the drivers for clinical response. So here we know that we have a molecule with well-behaved PK. So the higher the dose, the higher the PK, that's no issue. And we assume that the higher PK would translate into higher target engagement at the site of action and resulting in clinical response. It turns out not to be the case. And what happens in the end is that we realize that in our patient population, given that it's a late line relapsed refractory non-Hodgkin's lymphoma setting, all of our patients had received prior therapy that contains rituximab, so which is another CD20 monoclonal antibody. And rituximab binds to overlapping binding epitopes as MOSAN, which is shown as the 2H7 denotation on this slide. And therefore, it can compete for target engagement on CD20. And since patients all receive very different prior therapies at very different time, they come on to our trial with very varying levels of residual rituximab at baseline. So no wonder there is no dose response or exposure response, because this large interpatient variability in residual rituximab, which directly competes for our target engagement, can play a significant confounding role in understanding of our exposure response. So essentially what's going on is illustrated here. So I mentioned to you before, MOSAN works by binding to B and T cells, and together that creates an immune synapse, which drives anti-tumor activity. And in the presence of residual rituximab, what can happen transiently is that rituximab binds to CD20 and displaces mosunetuzumab's target engagement, and that reduces the synapse and therefore resulting in reduced anti-tumor activity. So knowing this pharmacology principle, the hypothesis, it allowed us to use mathematical ways to account for the impact of this transient competition on the exposure response. And in fact, we did something really simple based on principles of binding kinetics. One can derive at equilibrium the receptor occupancy based on the PK of MOSAN and rituximab, as well as the respective binding affinities, as shown in the equation here. And an interesting exercise, maybe perhaps for you all to try offline, is you can prove to yourself that CD20 receptor occupancy can correlate approximately linearly to the synapse level in this context, as long as the clinical dose is very low and at a subsaturation level. So in this case, it becomes a really mechanistic, insight-informed, and yet a very practical metric that we can use to understand our exposure response. So by using this very simple mechanistic concept of receptor occupancy, we swap the x-axis here to receptor occupancy instead of dose, and voila, we can see a very clear exposure response relationship that was originally perhaps masked by the large variability introduced from this residual rituximab. So this type of relationship is exactly what we needed to find the maximum efficacious dose and guide clinical dose decision. And an important strategic impact is that this also reviews the true therapeutic potential of MOSAN in an aggressive NHL histology context, where the estimated Emax values for ORR is about 70% and for CRR is about 40%. So it's really just a matter of finding the dose and the regimen to get to the therapeutic potential. And without this mechanistic insight and exposure response modeling, the exposure response relationship would have been masked, and we could be just taking the average across all dose levels and come up with the wrong understanding of what the molecule could potentially do. So this is my summary slide. So hopefully today I've shown you some examples of how mechanistic insights and modeling helped inform the clinical development of MOSUN. And through the quantitative systems pharmacology framework we built in collaboration with the translational systems pharmacology group, we designed the step-up dosing framework, which was proven now by clinical data to effectively mitigate cytokine release syndrome. And the insights really, thinking back, the insight really lies in the kinetics, where CRS risks changes with time, and presumably as a function of target kinetics, but also an intrinsic property of T-cell desensitization for cytokine release. And I've also shown you that the use of a model-based metric of RO, or receptor occupancy, just based on principles of binding kinetics, can be useful in understanding exposure response in a phase 1 setting to determine the maximum efficacious dose. And what I did not have time to go into today is really to put that into the context of disease kinetics. So I mentioned that the residual rituximab really only results in a transient competition with for target engagement, because as you can imagine, as rituximab washes out over time, eventually the competition is no longer there. So this time course really plays in where if you have a very fast-growing tumor, then this transient competition could result in clinically meaningful impact on response. Whereas if you have a very slow-growing tumor, such as follicular lymphoma or other indolent histologies of non-Hodgkin's lymphoma, then the time window to target engagement really is a lot more tolerable, and therefore a transient impact may not result in clinically meaningful impact on response. So I think kinetics here really plays an important role in understanding both safety and efficacy. I alluded to very briefly that at very low clinical dose and with low target engagement level, you can expect the receptor occupancy to approximately correlate linearly to the synapse. And in fact, at very low dose levels, we also expect a monotonic increase in terms of exposure response, and the bell-shaped understanding from binding kinetics in vitro setting in our case did not apply because through a properly designed phase 1 trial, you can stay in the range of still monotonic increase for exposure response. And eventually the dosing regimen we developed led to a broad therapeutic index, and that allowed us room to dose to maximal benefit risk. And hopefully all of the stories today link to the theme of the usefulness of quantitative pharmacology and mechanistic modeling. It really did play an integral role in MOSUN's phase 1 clinical development. And in fact, we were one of the first participants at the FDA's MIDD pilot, and Brendan Bender at this year's ACOP presented our case study. So I don't know, maybe some of you guys were able to make it to his presentation, but it really offers a great opportunity to gain early feedback and alignment from regulators. And there's a lot of great scientific discussions on model-based strategy, and importantly on its applications in drug development. So with that, I really want to thank actually a lot of people, but in particular, the people who actually did the hands-on modeling of the work. Brendan Bender is the modeling simulations lead on mosunetuzumab project, but also Fei-Fei Li for generating a lot of the exposure response relationship we talked about today, and Monica and Raj from the translational systems pharmacology group. and our project team, management, patients and investigators, and also the Certara webinar organizers for this opportunity and helping with all of the technology support. Thank you so much.
Thank you so
With
much.
Go ahead.
Thank you so much, Chi and Pete, for that excellent presentation. Good to see you both. Our audience, we encourage everyone to submit their questions to Pete and Chi in the questions panel, and we will answer as many as the time allows. So Chi, I've got a question for you. When compared to step-up dosing, flat dosing showed increased exposure. So instead of having 3 doses, as in the step-up approach, why can't a flat dosing strategy be considered?
So I think the choice between step-up dose versus flat dose is in its sensitivity to drive CRS. So flat dose, basically, you hit it hard directly at the target dose in order to get to the efficacy level you need. But at the same time, you hit it hard when the CRS or IL-6 exposure response is the most sensitive. So it gives you a lot less room to play with, if you will, in terms of therapeutic window. Whereas if you hit it a little gentler with a step dose when the CRS cytokine, pro-inflammatory cytokine is very potent, and then come a little bit later with the target dose. So the cumulative exposure that drives efficacy is still there to support your efficacy. And that's one strategy to decouple the drivers for safety from efficacy and therefore leading to better benefit risk. So I hope that helps answer the question.
Thank you. Pete, there's a question for you. Is there a specific software available to model and analyze preclinical and clinical data for bispecifics?
Well, I'm not aware of any software that is solely dedicated to, doing modeling of bispecifics. I mean, there are softwares that have, libraries perhaps of standard models incorporated. And I mentioned the SimCip biologics simulator, which has a extensive PK model for bispecifics. I think many modelers on the call will use more kind of general modeling software that the pharmacometricians and QSP modelers will know to build their own bespoke models. And I suppose the IO platform that I mentioned sits somewhere in between, where we can build bespoke models for the bispecific drug inputs, but couple it to a more kind of general IO QSP platform.
Thanks, Pete. There's a question for Chi. Is it possible to adjust the dose of the bispecific to the measured IL-6 levels in individual patients to reduce the risk of cytokine release syndrome to 0?
Oh, that's a great question. I think that's definitely something we're keenly interested in. I guess the first step in terms of what we could help as a quantitative pharmacology community is to identify that quantitative relationship. So in terms of driver for CRS, so in addition to perhaps IL-6 level, how you integrate it in a multi-dimensional way, including baseline factors. And in our case, it's also clearly a function of PK or receptor occupancy, if you will. So if you can derive a multivariate model that describes CRS rate, then you can start actually playing with positive predictive value, negative predictive value, and come up with a practical cutoff that you like to use. And I think it's possible. It just requires some quantitative insights. In addition to that, I'd say point of care assays, which is really rapidly picking up on its capability and the real-time measurement of IL-6 outside of site of administration, that would really make that a reality. So I guess it's a technology and analysis, and I think it's possible. You need to put some concerted efforts into that, I think.
Thanks, Chi. Pete, someone wants to know, have bell-shaped dose-response relationships been observed, or are they at present more a theoretical model prediction?
Okay, that's a great question. Or maybe the question is, are they real or artifacts or just dreamt up by enthusiastic modelers, right? I've worked in drug development for a very long time, and in all honesty, bell-shaped dose response curves always raise their head at some point. And in my experience, until recently, they were usually, if not always, somewhat of an artifact, either just an artifact in the data, or there was some kind of easy explanation that maybe some safety kicked in at higher doses and hence the response was kind of modified. But as a true kind of underlying pharmacological mechanistic shape, I never really saw examples that really were compelling until bispecifics. So I think the answer to the question is yes or no. Actually, I think they are real. I don't know what the question was. Are they artifacts or real? But they are real, I think. I mean, first of all, because it just makes perfect pharmacological and scientific sense. And the models that we showed, I think, are relatively straightforward and really capturing the key pharmacology of bispecifics. And it just makes real kind of common sense that you would expect to see bell-shaped curves. Also, working with various partners, we have seen actual experimental data, both preclinical, in vitro and in vivo, as well as clinical observations, which are obviously sometimes a bit harder to interpret given the perhaps narrow dose range and the variability that she showed. But I certainly have seen them, and I believe they are real. Chi, would you like to comment on that? Would you agree? Or
yeah, I think it's definitely possible given the pharmacology plausibility, and I think, but then that puts more owners, I guess, on in our clinical pharmacology community to really think smartly about those findings. Because I do think if you push this as any other oncology drug development where you just keep going until intolerable safety, then you might be even at the risk of efficacy. So.
Great. Looks like we have time for one final question, and it's for Chi. Someone wants to know whether there was a disconnect between the in vitro KD versus in vivo estimated KD based on the clinical data.
Oh, that's an interesting question. So let me think about how to answer that. So first of all, the in vitro KD value itself, as you're probably aware of, has multiple caveats, and you really have to know your assays to understand how you translate that in an in vivo setting. And in our case, really is the relative KD that we worry about, because it kind of gives you the strength of competition between rituximab and Mosun. So in our case, we actually ended up, because knowing that our KD measurement is subject to variability, in our case, we ended up doing a sensitivity analysis. So we looked at actually a range of KD values, and it turns out that when you already have the efficacy on the y-axis to anchor your exposure response, it actually doesn't matter what KD value you use in terms of dose determination. So what it essentially did is it just shifts the curve with different potency depending on the KD value you use. But let's say a lower relative KD for rituximab will result in a higher competition, and then that would result in a shift of the curve to the right. At the same time, the lower KD also give you lower Mosun, dose RO relationship. So it ends up shifting the dose RO versus RO response relationship in the same way, so that actually doesn't matter in terms of dose determination. So I guess that's a long way to answer your question of how we addressed it, but I think that the actual KD values really could vary quite a bit if you use different assays and if you use model-based approaches. In the end, in our case, it didn't matter.
Great. Thank you so much, Pete and Chi, for that excellent presentation. And thank you so much to our audience for all their excellent questions. We've just got a few points before we wrap up. The next webinar in the series will be next Wednesday. Dr. Jaap Mandema and Dr. Ojen Cox will present model-based meta-analysis for comparative efficacy and safety of therapeutics for COVID-19. And then the week after that, Dr. Daryl Nix will present Enhance Your Drug Development Capacity with ClinPharm Study Execution and Operational Support. You can register for all webinars by visiting certara.com. Next slide, please. On behalf of Certara, I would like to thank you for attending this presentation. This concludes today's webinar. Goodbye and have a great day.